My name is Camryn Young. It is August 11th, 2025, and this is my video demonstration on the experiment we are doing with the DNA extraction. So DNA, otherwise known as deoxyribose nucleic acid, is the building block and blueprint of life on Earth. It is a phosphate group, a sugar group, and a specific nucleotide, whether it's adenine, thymine, cytosine, or guanine, and they form together in two strands that wrap around each other to make a double helix structure. Now, this structure, while small, invisible to the human eye, and seemingly insignificant, carries hereditary information, directs protein synthesis, can replicate and fix itself through repairs, and do many, many other very complex things that make us who we are. And it's seen in all forms of life, and in the following experiment, even though most DNA can't be seen unless through an electron microscope or similar events, we are going to extract it from peas and make it visible to the human eye through a series of steps. So here we have our halved peas soaking in some salt water. The night before, we had them soaking overnight in distilled water just to soften them a little bit, and now that they are softened completely, we have it in this half full distilled water and salt combination. This is important because DNA's phosphate backbone is negatively charged and can be soluble in water when interacting with the charged DNA. So we want to neutralize it a little bit with salt, which is positively charged. So it will interact with the DNA and neutralize some of that negative charge and make it less soluble in water. So after the peas were crushed by the mortar and pestle, they were strained into this drinking cup, and half a bottle of the dropper dish detergent here was added to the drink and carefully stirred in. This detergent is added because it will break down the cell membranes within the pea cells and allow the DNA to officially come out so it can be observed. So once our solution has cooled for 10 minutes in a refrigerator and has been moved to this test tube, I have it sitting in this cup just because it's a round bottom flask and I don't want it spilling. We are going to add this meat tenderizer or papain powder. Papain powder is important because papain is an enzyme that's found in the papaya, and it works in cleaving the peptide bond of proteins that can interfere with DNA extraction and analysis. So when we do this, it breaks those proteins into smaller fragments and improves the purity and the yield of the DNA extraction we will get. So now that we've added the papain powder, it is now time to add equal amounts of ice cold alcohol gently to our solution. This is primarily used to precipitate the DNA, which makes it visible and more easy to observe in our experiment. The reason why we use it as cold is it will help kind of protect it from degradation, because the cold temperatures will slow down the activity of DNases, which minimizes the damage during this extraction process. It will also reduce the solubility, as DNA has a lower solubility in cold alcohol versus warm alcohol. So after letting the solution sit for about a minute, we are getting these stringy white filaments of DNA forming between the alcohol and the official solution. It's definitely a little less than what I was expecting, but at the same time, DNA normally isn't visible to the human eye. So the fact that it's clumped together enough to be visible at this amount shows a pretty extreme change, because that actually is a lot more than I expected to be visible, because DNA is so small compared to what we are seeing right here. Overall, I think it was a really great experiment and was a beautiful show of DNA extraction. Thank you very much.